And these are my two co-authors, Muñoz, who is my PhD student, and he finished his master's degree January this year. And the results I will show today are his master's thesis. And Reyes, who is a professor at my university, and she helped us with the sperm analysis. So we come from Puebla University in Mexico, where I started an evolutionary ecology lab 5 years ago. And we mainly study sexual selection in guppies. Today I will talk about the relationship between sexual selection and parasitism. As you know, parasitism is the relationship between two species in which one of them benefits at the expense of the other. Sometimes without killing the host organism, but sometimes leading to death. And I'm sure all of you are familiar with this picture. Marlet's cricket dying horribly after being infected. And what about parasitism and sexual selection? So parasite-mediated sexual selection has traditionally been linked to the evolution of extravagant male traits such as coloration or courtship. So we have a good understanding of how parasitism affects precopulatory traits in males. If you look in the literature, parasitism and sexual selection, what you find is that most studies have looked for this correlation, so parasite load and male ornament. And what most of the studies have shown, but not all of them, is that the more parasites a male has, the less expression of the ornament it will show. And again, I'm talking about ornaments here, but what about postcopulatory traits? We know that postcopulatory traits such as sperm quality are very costly to produce, but less attention has been given to understanding how parasitism affects sexual selection after mating. A lot of studies, what they have done is they have experimentally activated the immune system, and then they have measured how this affects sperm quality. A few other studies have used natural parasitism, and they correlate that to a few sperm quality traits. But these studies have been conducted in farm animals, and some of them have a veterinary perspective. Not all of them have an evolutionary or behavioral ecology perspective. And I have some concerns about our literature in this topic, because different quantification methods have been used. So one study has measured, for example, sperm number, but not sperm viability, and some studies have measured gonads, but not sperm mobility. So we have different quantification methods. The other thing is we have a lack of experimental infection. So authors have just used natural variability, which is good. But we need stronger methods to test hypotheses. And then there are some contradictory results. So for example, in the wild goat and the Arctic char, mass of gonads and the wild goat will go down after infection as well as sperm mobility, but in the Arctic char it will go up, both traits. So we need more experiments trying to see what happens between parasitism and postcopulatory traits in males. So that's how we came to this question. How does parasitism affect male sexual behavior and sperm quality? And we use the guppy, a very well-known model species. As you know, it's one of the most studied live bearers with strong sexual dimorphism. Males are smaller and they show this beautiful coloration, and females are pale and larger than males. They have internal fertilization, and the males have this, as any Poecilia, they have this anal fin that is modified and that males use to inseminate females. This picture here is the sperm bundles, so sperm are packed in these bundles, and then you can break the bundles and study the sperm inside them. And females can store sperm up to 9 months, which means there's strong sperm competition in the system. So these guppies use two different tactics to inseminate females. One is courtship, so this male is courting this female here. And the other one is a sneaky behavior or forced copulations. So where the male gets close to the female trying to insert thrust the gonopodium inside the female without courtship. The most studied parasite in this system is Gyrodactylus. This is a flatworm, ectoparasite. It's very interesting because it has a peculiar reproduction. It works like a Russian doll. So you have a juvenile flatworm that has the following generations inside it. So you can see the grandmother, the daughter, and the granddaughter in the same system. And that's bad for guppies because that means the parasites will increase quite quickly in a population. The prevalence is high, it's like 75%, and guppies will have like between 4 and up to 30 parasites each. But 30 parasites means death for guppies. So it also has economic implications as well. So Gyrodactylus and guppies have been widely studied, as I said. We know that these parasites affect body condition, immune system, swimming performance, sexual behavior in males, male coloration, foraging ability, and social interactions. But nobody has studied sperm yet. So how we did the experiment? We experimentally infected males. So this is Luis doing the work, the lab work. This is one female. This is the mouth, and as you can see, it's full of worms. What we did is we bought commercial guppies, and we looked for Gyrodactylus. After that, we grabbed one parasite, and then we infected more of our Mexican native guppies that were collected in Morelos state. And once we had a lot of infected guppies, what we did is we brushed the worms, and we put 2 worms in one container and 1 male. And after between 3 and 5 minutes, we checked the males at the microscope to check if they were indeed infected. And in 100% of the cases they were, to our surprise, because we thought, well, these worms are not supposed to swim, but we got it. So this is one worm here and the other one here. They move, so it's easier to check at the microscope. So that's our infection methodology. So what's the experimental design? We had 2 treatments, the control males that were only placed with water, and the parasitic males or the infected males. We compared sperm samples before and after treatments. And the infection duration was 7 days because our population didn't have Gyrodactylus, which is very strange. But we sampled the population for 5 years, and we never saw Gyrodactylus there. And they die very easily. So after 8-10 days, 60% of the males infected die if you don't treat them. So basically, we have healthy males here, and we collected our first sperm sample. Then half of the males were allocated to the control treatment or the parasite treatment, and then we collected a second sample. And although male behavior and parasitism has been well studied in this system, we also measure behavior, and we did it 1 day after the treatment. So the males were infected, and 24 hours later, we also measured their behavior. And the way we did it is we placed 1 male with 2 females, and we measured courtship and forced copulations, as well as gonopodial swings, which means that the gonopodium, the anal field, they can move to one side or the other. And we don't really know why they do it, but it seems that is to place the sperm to make it ready to inseminate females. And also association time with females. And regarding sperm analysis, we measure everything that people have measured with sperm in in guppies. Obviously, we follow the experts in the topic, and we measure quantity, and then you can experimentally extract sperm in guppies. We measure bundles, frequency, and also sperm concentration in a standard way. We also measure quality, which is mobility, viability, and velocity. And velocity has different measurements, so we conducted a principal component analysis to see which ones were the ones that we wanted to study. And this is obviously after data collection, so these two are the important ones, and I'll talk about them later. We also measure morphology, and it is sperm length and flagellum length, the most important variables as well. So what we found regarding behavior, in accordance to previous studies, we saw that infected males show less mating effort and also less gonopodial swings. And also in accordance to the literature, parasited males spend more time close to females. So this is something that has been shown before as well. What's new is what we found about sperm quantity. So if you see before the treatment and after the treatment, both control males and infected males show the same number of bundles, okay? So there is no change in bundle number in our experiment. Oh, sorry. And the same was for sperm concentration. So if you measure the number of sperms in the samples, they don't change with parasitism. But sperm mobility and viability were strongly affected by the parasite. The average path velocity and the curvilinear velocity was also affected in infected males. So it's strongly changing the sperm quality, and remember there's a strong sperm competition, so velocity should play a role in sperm fecundity as well. Oops. And then finally, regarding morphology, we found that results that we don't know how to interpret very well. Sperm length and flagellum length both decrease in both treatments. And if there is someone who can help me explain this, I would be happy to talk to you after my talk. We think the explanation is that being as isolated is affecting both treatments and reducing the length of the sperm. So in conclusion, sexual behavior was reduced in infected males. When we compare both sperm samples, spermatophores and sperm number were similar in both control and infected males, but infected males show less sperm quality in terms of viability, velocity, and motility. So for the first time, we showed that parasites not only affect precopulatory traits, but also postcopulatory traits. And the interesting thing is, at least in our population, it doesn't seem to affect the first part of spermatogenesis, so it's just later stages. So I would like to thank my university for funding to conduct experiments. I mainly work with undergraduates, and they are the ones that help in the lab a lot. And also, I would like to emphasize that coming to the conference is only possible through the travel grant, so thank you to the organizers for that. And if you saw your picture, don't get upset. Thank you. That's a good question. After the second sample, after the behavioral samples, the behavioral test, we measured the number of parasites in each male, and it did change. We also conducted the same analysis using a continuous variable, so it's a parasite number, because as I told you, it did change. It does change after time. So we got up to 14 parasites, and we didn't only with velocity and morphology, but we found positive correlationship. A negative correlation between parasites and sperm quality. It's funny because a lot of studies use two treatments to test parasitism. So infected or low parasite load and high parasite load. And it would be interesting to compare why we don't use continuous variables for parasite number. So we did controls for parasite number, and when you run the test, the models, they don't show significance. No, it has to be like no parasited versus parasited.
Do females acquire the parasite during copulation?
Yeah, that's Louis' PhD thesis. We did when we use the behavioral test, we used non-infected females, and I was sure they were not going to get infected, but I told him put them apart, and then to our surprise, they got infected. So that we are going to see how the sexual conflict with parasite, if I get the grant.